Today, gives me great pleasure, in fact, to introduce you to Associate Professor Jessica Ma from the University of Queensland. And she's currently a group leader at the Australian Institute for Bioengineering and Nanotechnology. And she leads a computational biology group that investigates how variability in the genome contributes to the regulation of diseases like cancer and phenotypes of pluripotency in stem cells. She received her PhD in biostats from Harvard in 2008, so that's very prestigious. And she was a postdoctoral fellow at the Dana-Farber Cancer Institute in Boston and an assistant professor at the Albert Einstein College of Medicine in New York. So some very lovely postdocs there and lots of good experience. And she's relocated back to Australia as an ARC Future Fellow, and she arrived last year, so we're very thrilled she's back here. She's also received special awards including a Fulbright Scholarship in 2003 and a Metcalf Prize for Stem Cell Research from the National Stem Cell Foundation of Australia in 2017, and La Don H. Schumann Award for Teaching Excellence at Albert Einstein College of Medicine in 2017. So she's got lots to offer, and we're really looking forward to hearing about her topic, which is title. Oh, it's right in front of me. Sorry about that.
It's on the screen. People can read it. It's okay.
Yeah, it's Leaving Statistical Shapes in Genomics, Looking Beyond What's Normal. And we now have 13 people, and one of them is actually tuned in from Oslo. So we're very excited about having a guest from overseas. So welcome, Jessica.
Thank you so much, Miranda. It's a really wonderful opportunity to present, especially in this audience. So thank you to all our visitors and guests. So I'd also like to mention some people here from my group, including one of my PhD students who's dialing in from China. So it's really great to see everyone, and especially just to be able to, as I said, give a talk in this kind of environment. So my name is Jess, and I'm actually not a stranger to the Statistical Society. And thanks to the internet, I actually dug up this old newsletter that was floating around. This is from November 2002. And I'm not sure if you might have this in your collection if your records go back this far, but I was really fortunate. My first ever talk that I gave in my research career was actually as part of some of the young statisticians events that were occurring. So I remember going to QUT, being an absolute nervous wreck, and presenting my 15-minute talk on research that I'd done. And one of my mentors here at the University of Queensland, Gordon Smythe, was absolutely instrumental, I think, in just giving me that confidence to go for this opportunity. So that was one place where I was able to really develop. And then over that time, over those years, there was obviously an opportunity to present at a branch meeting, which is what this article refers to, but also the society supported me to go to an annual meeting. I remember in Canberra, I believe, which was so exciting because as an undergraduate, I wasn't really in the habit of going to conferences. And so that was hugely meaningful. And even now that I'm back in Australia, they supported one of my students, Melindri, who I'll talk a bit about her work later on. So to go to also the annual meeting in Canberra. So there's been so many opportunities and lines of support that have been extended. So in a way, it feels a little bit like coming home to be able to present again. It would have been great to be in person, but online formats are also really great too. I'd like to highlight too that this article was written by Peter Baker. So I think it's also an incredible effort that he's still leading the charge for Queensland for this particular branch. So kudos and thanks. And so I'd like to just start my talk very broadly. I know as statisticians, we come from different kinds of slices of science. And my particular slice of science is biology. So I think if we bring it all the way down, one of the jobs that we have in my field of research is to really understand how biology is working. And we do that through using different kinds of modeling approaches, but effectively one of the recurring themes is really to think about having a patient population from which we sample and measure a biomarker of particular interest. And so what we have then from that data is a distribution of this biomarker that's quantitative in nature. And if we're lucky, depending on what we're studying, we have some sort of cutoff or some sort of clinical significance that we can attribute to the level of this biomarker. And therefore, inferences from that point flow very clearly. Often the case, though, in biology, we're interested in contrasting two populations. So either a control versus a disease population, or something of that kind of sort. And so the idea is the same. We measure biomarkers. We then have two kinds of distributions. And really, the name of the game here is to try to figure out where is the separation? Is the separation between these two groups large enough to warrant significance, or is there really no effect given randomness and noise? And so in the kind of world that I live in, a lot of the data types that we're working with come from different types of molecules. So DNA, gene expression or mRNA, but also not to neglect the more traditional clinical variables like weight, height, or if we're thinking about cancer, grade of the tumor, or other sorts of measurements that we might take from pathology. So this sort of kind of captures the rationale or the sort of thinking that we have when we think about modeling biology. And so to be really specific, as Miranda mentioned, I've been away building my career and came back to UQ to start up my group nearly 2 years ago now. And really the focus of my group at UQ is to understand distributions. And so really, if you think about it in a very simple way, if we take measurements of, say, the expression of a single gene in a population of cells, we end up having this kind of density distribution here. And I'm not sure, can you see my mouse? Yes? No? I'll just keep using, yes, yep. Excellent. Yes, thanks guys, thanks. So I'll use it here. So obviously, as statisticians, we're all very familiar with this kind of density distribution. But I think it is worth really reflecting on this and what kind of information we can gather. So as you know, we can use these moment estimators to estimate the underlying population structure of the data. And I think what's really key is that in bioinformatics, really these methods typically focus on what's going on at the first moment. But as we all know, there's lots of information to be mined. And so a lot of my earlier work was really understanding what was going on at the variance. So looking at the second moment. And we've got some different papers that reflect on studies that have done to look at the value of studying variance of gene expression. We can go even higher and look at skewness, the third moment. And last year we had a paper working with high school students looking at what we can learn from modeling skewness instead of just looking at the mean. If we think about distributions and how the shapes might be changing, we have to then think about other features that may not necessarily be captured by the moments, but they're certainly related. So things like bimodality, I think, is really interesting. And there's a story I'll tell you about today that was published in PK et al. Last year, looking at bimodal distributions. And I think really in biology, what's important to not lose sight of is that modeling these higher moments are really interesting because we can identify the extreme behavior. And it's often those kind of outliers or subpopulations that are occurring where the most interesting biology is happening. So I think there's a huge push, and this is becoming more recognized now, but certainly there is a huge push for going beyond average trends and really looking at what some of these higher moments can tell us. So this is in a way where some of the title of my talk is coming from in terms of looking beyond normal. I'll give you some other examples of that as we go on. So just to go back in time a little bit, the first kind of proof of principle that I worked on as a postdoc was really to ask this question about whether looking at variants of gene expression could help us understand how different phenotypes or diseases were regulated. And so I use this example here, which I know we're all very familiar with, using the T statistic, right? So the two sample T statistic, where often when we work with gene expression data, so this could be microarray or RNA sequencing, we think about two different populations. And we have, say, for instance, a disease group, a control group. And really what we prioritize when we look at the thousands of genes that are available is the separation between the difference in averages. So genes that are more reflective of defining differences between disease and control will have a larger difference in average. And that's a really sensible way to prioritize looking at genes of interest, but it doesn't permit looking at other kinds of behaviors. So for instance, these examples here where there is a difference in average, but as you can see, there's a very different profile and variability here for the disease group versus the control. And the more extreme example over here is the fact that there is no difference in average, but obviously, again, you can see the difference in variation. So these sorts of questions I find really, really fascinating. And this is this sort of question led to a paper in PLOS Genetics I published quite a while ago. So I think for whether you think about cells or not, I'd like to impress that I think for statisticians out there, it's a really interesting time to be asking these sorts of questions. And that's because we're at a stage now where we're able to sequence individual cells. So what I've got here on the right-hand side of the screen are these kinds of balls that are supposed to reflect different cells. And based on a lot of the kinds of assays or equipment that we've had in the past, we've really only been able to look at average transcriptomes. And by that, I mean all of the genes that are expressed inside a single cell. And obviously now we're in a realm where we can actually look individually at which genes are changing from cell to cell. And so we're able to get more of this complete profile. So we appreciate much more readily the fact that cells are heterogeneous. But from a modeling perspective, it means we can actually do a lot more meaningful data analysis. And so even though we've known about these concepts for a long time, finally, as with a lot of things, the technology is catching up. So I think it's a really great time to be thinking about these problems statistically. So I'll get started and talk about a specific project here. And this goes back to bimodal gene expression. So this idea that we can use bimodality to identify new regulators of cancer, and in this case, we're talking about breast cancer. So I just wanted to start off by, again, going back to introducing this concept that as statisticians, we often have to access lots of different kinds of different distributions. So we have the normal distribution, obviously, but we have other things too, like the Cauchy, the log normal, the Pareto distribution, which is more extreme, the gamma distribution, which is quite flexible, and also multimodal distributions, for instance, the bimodal. And so a long time ago, I had a postdoc who was interested in looking at the question about non-normality directly in human cancer transcriptomes. So we wanted to ask this question of whether all gene expression follows a normal distribution, or whether other distributions are actually quite appropriate too. And moreover, does every single gene that we're looking at follow a normal distribution, or is there variation when we consider all of the genes that are available? And so we use this resource called the Cancer Genome Atlas, which is basically open source data that looks at different tumor types. So we chose 3, acute myeloid leukemia, glioblastoma, and ovarian cancer. And you can see that the number of patients in these data sets really ranged from about 200 to about 600. And they built a classification pipeline to figure out which distribution shape fit a gene expression profile across these populations of patients. And you can see, hopefully, some of the tests that they used. It was sort of sequential, but we didn't try to force any of the distributions in, or any of the genes, excuse me, into a distribution. And so there was an opportunity for, if there was nothing fitting, then we removed those genes from the analysis. basically what we found was quite surprising. We found that 50% or more of the genes that were expressed have non-normal distributions. So the genes that are normal are in this gray bar here, and everything else, excluding the black, is a non-normal distribution. So this was data that we found using microarrays, which is an older technology. But actually for these data sets, what was really interesting is the same patients were profiled, but using RNA sequencing technology, which is a bit more advanced, a bit more precise. And so what we found was obviously differences in some of those counts, but while there were increases in the normal distribution component, the non-normal component certainly did not shrink to zero. So we found that was quite interesting given the prevalence of the normal distribution, especially in microarray data. And it's sort of interesting to see just the distributions or the counts in these different distributions. So a way we attacked this was to then focus really on, what this bimodal component could mean. And so I was really fortunate to have a PhD student in my group, Daniel PK, who is now just started out resident fellow at UCSF. And basically what we wanted to do with this kind of data was look for bimodal relationships. So you can imagine that we've got two different populations reflecting each of these modes. And we know already, due to different kinds of indices like the bimodal index out there, that we can model these bimodal distributions quite easily using Gaussian mixture models. And so these are just some of the parameters that came up for one of the genes that we found in the breast cancer cohort for this data. What we can see then is how do we extend this to be useful to oncology, to understanding cancer biology. And so here we basically use these models to fit what we're defining as an oncogene candidate. And so these genes are highly expressed in tumors, but in such a way that they're expressed in only a subset of the patient population. So for this kind of data, I should also mention that what we have is a set of tumor data from the patients in the cohort. And from those same patients is a set of adjacent normal samples. So these are biopsies that have been taken from the same patient, but they're adjacent to the tumor and non-tumorous as far as pathologists can tell. So what we've got is a really good kind of disease control setup here. And the idea of how we wanted to use these mixture models was to look for instances where there might be bimodality in the tumor profile, but unimodality or unimodal profiles for the adjacent normal, and such that the expression of this particular oncogene candidate was much lower in the adjacent normal compared to the tumor sample. So this is exactly the kind of cartoon that we were theoretically looking for. And what we did essentially was fit models around these kinds of assumptions to detect them in the data. And this is just an example of real data that we found using a gene that fit these sorts of criteria. So we'll go into this in more detail in terms of what we found, but essentially this is our workflow. We used about 110 patients from this data set. These are the clinical characteristics, and essentially they're just the different steps. As anyone who's worked with publicly available data, especially large data sets that have many different kinds of clinical and also genomic components, you'll know that there are a lot of pre-processing steps as well, quality control, just to make sure everything checks out as there is robust and available. And so this is also reflecting some of those different steps too. But essentially, if we go through some of the approaches, we came up with this selectivity index, and it's really there to separate or identify, that separation of the subgrouping of overexpressing tumors. So we're trying to find this kind of separation here between this sort of group. And you can see that for high SI, you've got this really beautiful sort of separation between the two groups in the tumor cohort. For lower SI, there's less of a separation. They're sort of just sitting all on top of each other. So we use this index to really rank or prioritize those genes that were meeting that criteria more cleanly. We then also wanted to just figure out which were overlapping here. So basically how overlapping was the lower mode with the unimodal normal expression. And so that's all brought together in this Oncogenes score. And basically we were able to select a cutoff just by quantiles and prioritize our top 5 candidates. So these are the profiles for the 5 candidates that met our screen. And you can sort of just see from the densities and the histograms that are here, just how they fit or how close they are, at least to this kind of theoretical profile that our indices were built up around. So that was really exciting to know that these kinds of profiles do exist in the data. And then we went through a series of analyses to figure out, well, what does that now tell us, right? It's one thing to find these genes that meet this profile, but how are we informed or updated in knowing that these genes exist? So these are the 5 that were in our final set to look at. What was exciting was that 2 of these had already some sort of prior role that was implicated in oncogenesis, and specifically CBX2, which is part of this polycomb repressive complex. So that's quite exciting. We applied Oncogenes, which is our software, to fit these distributions, find these particular candidates. We did compare that to some other kinds of analyses, namely looking at outliers to see if like an outlier detection method would recapitulate the results that we were seeing. It really didn't. And lemma, if you're familiar with, is basically a way to look for differentially expressed genes. So which genes are different from tumor versus normal. And again, really, they were not highly ranked at all using this kind of software. So we think that our particular approach is identifying novel candidates that you otherwise wouldn't have gotten with the standard sorts of approaches. So one thing we tried to do was really make sense for how these different genes were regulated. So given these bimodal relationships, could we predict what sorts of features might tell us whether you're more likely to have a tumor that ends up in that higher expressing mode versus the lower expressing mode? And for that, we used LASSO to basically do, to do ridge regression and come up with this predictive model. So if I just jump to the results, I can show you. So for each individual gene, we fit one of these LASSO models using these different sets of features that were available. So some of these were technical in nature. They referred to sorts of potential batch effects that we wanted to correct for. And so those reflect, location of the tumor in terms of hospital or sequencing plate. But also there were clinical variables that we had to take into account. So things like race, stage of the tumor, survival, age of the patient, and percent of tumor cells in case that was an influencing factor. For the genetics though, there are other expression-based metrics that were really important to take into account, including things like copy number variation. So was there more repeats found in the genomic DNA that could have contributed to being in this upper or lower mode? And then finally, we also wanted to include DNA methylation. So this can occur in different places in a gene, and basically the consequences of that functionally are a bit different depending on the location. So that's why it was split up across these different categories. So you can see that across the 5 different genes, they had varying degrees of success in fitting this model. So the AUC was really good for some of these genes like CBX2. But for others, like this EPYC, it was really no better than random at 0.5. So we think that that really reflects the biology, the fact that these genes all do different sorts of things. And you can see also by the coefficient ranks, the priority of some of these coefficients is really quite different depending on the gene that we're looking at. So the CBX2, the most strongest coefficient was the intronic CpG methylation, where basically we saw this negative model coefficient that is really predictive then. And so when we look at the actual data, this is the DNA methylation. It's reflected by these beta values. So the higher, the more methylation here. And so because it's a negative coefficient, we see more methylation in this lower mode, whereas the more highly expressing mode is associated with lower levels of methylation. So that's one way to sort of break down this information to predict which patient, based on these other sorts of features or characteristics, might be in one mode versus another. We wanted to extend this out and understand what could it mean also for survival of the patient, and whether this was actually indicative of different rates of survival. When we look back at the patient population that we had here, if we break it into patients that were in the lower mode and the upper mode, we do see a significant difference between the survival curves for these two groups, which is interesting. If we go back as well and look at the aggressiveness of this molecular subtype, what we used to do this was a computational classifier, which takes genes that have been known to be important and allows us to basically put the different patients into these subtypes. So you know that breast cancer is defined by different molecular subtypes, and these can have very different clinical outcomes and degrees of aggressiveness too. And so once we look within these different groups into which patients fell into the high expression mode or the low expression mode, you can see that the proportions really track with the aggressiveness of the tumor. So what's interesting about this is that when we use this computational classifier, it was made up of a bunch of genes, but the gene that we found, CBX2, was not part of that. So it sort of opens the door of perhaps CBX2 might be useful as a way to identify or even subtype aggressive breast tumors. Finally, what we wanted to look at too, using other kinds of data, was to know what was the expression level of CBX2 in a population of healthy people. So for this, we used a resource called GTEx, which is Genotype Tissue Expression Portal. And basically, we were able to look across all the different kinds of tissues in healthy adults. So in this case, it's 53. And we see that CBX2 is really only highly expressed in the adult testes. That's this group here. So for a potential target for breast cancer patients, this is actually a really good thing. We know that under basal sort of circumstances, CBX2 is not highly expressed normally. So that makes it an excellent target. Just the final thing we wanted to do here was to look at in cells that are cultured in a dish, what happens when we manipulate CBX2, specifically what happens when we knock it down. And what we saw in these breast cancer cells, this was a human breast cancer cell line called MCF7, if you're familiar with your breast cancer cell lines. And basically what we saw was a much slower growth of these cells when we knocked it down using this siRNA. So that's the green profile that you can see versus the control. And this was done in collaboration with Cristina Montagna, who's my colleague at Albert Einstein College of Medicine in New York City. So this is really encouraging, even though we're moving away from looking at patients now, but to see directly that results from our computational predictions were actually validated in the lab. And this is often how we use our statistical models to identify potential hits that we can then take back to our colleagues in the lab to get more information. So I think for this part, all I'll say then is we found a few different really interesting things that really started with the premise of whether bimodality in this kind of data was actually helpful for identifying new targets for cancer biology. And this software is packaged up in Bioconductor in R. So if you're familiar, this is freeware version, is publicly accessible. So feel free to take a look. We also had this published last year in the British Journal of Cancer. So if anyone has questions as well, I'm always happy to take them during the talk, at the end of the talk, or at another time too. But what I wanted to highlight in this talk summary so far really is this looking at heterogeneity and gene expression, looking at those different kinds of moments, not just average trends, is really informative for understanding how phenotypes are regulated. So for instance, we've used this example of cancer. And I think to go one step further then and think about the distribution shapes themselves is really important for understanding what those new regulators could be. And so CBX2, the one we found in breast cancer, I think is a really good example of that in action. And the next thing I want to talk about briefly is this idea of taking it one step further and capitalizing on the availability now of all these single cell data sets to model heterogeneity, specifically to understand whether we can get insights into the aging process. So aging is an area that's gotten, I think, a lot of activity, but I think it really lends itself to this concept of heterogeneity. I mean, anecdotally, we all know that person to person, we all age differently. And at the molecular level, while there certainly are some hallmarks that dictate that process, there is a lot of noise, there's a lot of variability from organ to organ, from cell type to cell type. And I think this is a really great place where we can use these kinds of statistics to understand heterogeneity directly, really to get more insight into how we age. So first off, I just want to introduce some of the work that one of my PhD students, Hui Wen, is doing. And I believe Hui Wen is here with us, so she can answer all your questions. But really, the purpose of this study goes back to the idea in statistics that we have lots of different ways to measure a specific property. So we have lots of different statistics, and as we know, they all have their pros and cons. And as well, sometimes their performance is highly data specific. So with looking at gene expression variability, it's really no different. And when you're looking at what are the tools that people are using out there, there really is a whole spectrum. And what we noticed is that usually people pick up one and run with that, but there isn't really a comprehensive investigation as to which statistic works best, on what kind of data. So there are different sorts of ways single cell data can exist. There's also different sample sizes, different cell types, and different sort of levels of technical noise that we might expect. So how do these different statistics that are based on different kinds of pros and cons, how do they perform on these different kinds of data? And also, as with anything, what conditions or expectations can we expect this performance to behave under? So Hui Wen's undertaken this massive comprehensive study to basically help us understand which metrics are the best for measuring gene expression variability. And so just to give you an insight into one of the ways we're asking this question, as you all know, the structure of a data set can really influence some of these statistical measures, and often unproductively. And for single cell RNA sequencing or gene expression data, one of the features that we know about this data type is that there's an overabundance of zeros. And that can be because we're sequencing genes inside a cell. So many of these genes might be turned off. So there's a biological reason for why we see a zero. At the same time, we're dealing with such small quantities of biological material that we might not just have enough to hit that sort of detection limit. And so we would consider that a technical zero, where it's just zero because there's no way to get signal for that particular gene. And so either of those reasons means that we end up with lots of zeros in the data. And as we know, zeros are a bit of a special case, and they can really make calculation of some summary statistics quite challenging. And so a prime example here is when we think about the coefficient of variation. So this is the standard deviation divided by the mean. And you can see that when we consider how many zeros a gene has in a population of cells, the more zeros we have, the higher the coefficient of variation. So the statistic is heavily correlated with the number of zeros, which isn't ideal because it means that if we were to use CV as a measure of variability, our sense or our measure is therefore influenced purely by the number of zeros in the population. If we look at another metric, so this is the local coefficient of variation, it's a smooth version of the CV. We can see that as the number of zeros increases, the relationship still is independent. So although it looks messy that there's no relationship here, that's what we want to see actually, because each of these dots reflects a gene, and for various different reasons, for a population of cells, we'd expect genes to have high degrees of variability or low degrees of variability. And the whole point is to be able to have a metric that can tell us which is which. So based on this particular criteria, what we've done is we've ranked the different metrics that we're looking at from the worst to the best performance. And Hui Wen has a metric that she's used to summarize performance. But basically 1 is our worst performer, like the CV in this case, and 10 is our best performer. So in this case, it would be the LCV. And so really, because there are lots of different metrics and because there's lots of different ways to evaluate the utility or the performance of these metrics, what she's done is come up with a comprehensive heat map that reflects how well each of these metrics was evaluated for these different metrics. So basically for each of these, like the CV, you can see that based on the different criteria that we used, it did quite well in some, which is this blue sort of coloring here. But for others, it also did quite poorly. So for instance, as we saw here, for the number of zeros, it didn't rank very well because of that correlation that we saw. So obviously, the most important thing is who won. And so based on our overall tests, we see a lot of promise for this particular CV-based metric, but nevertheless, it's got some modifications to it to iron out some of the problems or some of the issues that we saw with CV. But overall, it's got really strong performance across the different criteria that we tested. Why is this important? This is important because what we want to do now is use this as a way to understand how the transcriptome is changing with respect to variability during the aging process. And for that, we're using this resource. I've hidden the most important part. I apologize, called Tabula Muris. And Tabula Muris is a publicly accessible resource where they've sequenced at the single cell level organs from different parts of the mouse. So these are the types of organs that are available, and you can see how much sort of cells there are, which is pretty cool. And what we want to do is really ask these sorts of questions by taking mice from the earlier, so the younger time points at 3 months of age to 24 months, so a much older mouse. And we want to use this metric to understand how variability is changing from young to old for different kinds of organs. And within those organs, there are different cell types. And so how those cell types are behaving in terms of which ones are getting more variable or less variable, and what are those genes and pathways that are experiencing those sorts of extreme changes. I think that's what's really going to be interesting. In thinking about contrasting these different organs, is there a universal kind of signature in terms of which genes or pathways show changes in variability? or are there just ones that are purely very organ specific? So this is fantastic work that Hui Wen is doing, and we're excited to see where that is going to go. So what I wanted to talk about next, before I run out of time, is a project that one of my PhD students, Melindri, who I mentioned at the very beginning, who was lucky enough to go to the annual meeting in Canberra last year. So Melindri is working on a really interesting project that's looking at different distributions in single cell gene expression data. And this goes back to the study I was telling you about, where we were looking at the cancer genome and trying to understand, you know, which genes were gamma distributed or log normal distributed. Basically what we're doing now here is the same kind of question, but for single cells. And what's really fascinating here is that because of the overabundance of zeros, what we often see is the prevalence of these zero inflated models. So these are models that allow for a continuous component, but also a zero distribution. So basically the representation that there are zeros that are part of this distribution. So these are zero inflated Poisson and zero inflated negative binomial examples that Melindri has drawn here. And also, obviously we can't ignore their individual one part counterparts, which is the Poisson and the negative binomial. And actually when we think about single cell RNA sequencing data, it's worthwhile pointing out that most of these distributions are assumed to follow a negative binomial using these tools. The other thing I want to highlight is the same kind of assumption that we had before, where regardless of the gene that we're dealing with, there is the assumption that that gene will follow the same distribution. So what we're doing differently here is we're allowing for the fact that genes might have different distributions, and if so, what are they? That is really what's interesting. So Melindri, I was really lucky to have Melindri join my group. She comes from a background in statistics and math. And so what she did was develop a framework to set up the testing environment to determine which genes are different in their distribution. And so I think all the formulas come up properly, but basically she steps through these different kinds of tests, starting with the KS test to determine whether the genes are actually belonging to this family of zero inflated negative binomial distributions. And then the idea to fit these different models and determine of these candidates which is the best fit. So that's really effectively what we're trying to do, using different statistics to guide us along the way. And how this is happening in action is using a data set that I generated in collaboration with Professor Nir Barzilay, who's director of the Institute for Aging Research at the Albert Einstein College of Medicine, along with one of my PhD students who we co-supervised, has graduated. So my apologies, that should be Dr. Ameya Kulkarni. And basically we looked at mice who were either young or old, and also old mice who had received a drug called metformin. So this is a drug that's prescribed for type 2 diabetes, but it's caught a lot of attention around the world because it does have anti-aging properties in model organisms. And from these mice, we were fortunate enough to be able to take two different kinds of tissues. So we took adipose and we took muscle from these three mice from each group. And so what Melindri has done here is set up quite a complicated framework, but it's really quite straightforward. It's just because there's so many different kinds of analyses and variables, but effectively we're looking at different pairwise comparisons. So old versus young, for instance, for each of the tissues. And we're interested in understanding which genes flip from one distribution type, whether it be zero inflated Poisson, to a different kind of distribution, whether it be the Poisson counterpart or a different kind of zero inflated distribution, for instance. We want to contrast that to what is traditionally done for this kind of data. So looking at differential expression, what is just shifting in mean or average. We want to understand what that tells us about aging and also what sort of biological pathways do these genes that do change their distribution, are there specific trends that that are interesting to see? So basically just oh, someone's drawing on my slide, which is great. Basically just to cut to the chase, there's a lot of work that's done here, but we're sort of in the process of consolidating the results. But what I will tell you now is that when we look at how well the negative binomial does, it's really interesting because for adipose, we see an overwhelming majority of genes that are explained by the negative binomial distribution, with less representation across the other categories. But for muscle, it actually comes back more towards 50%. So the other distributions are actually are a lot more well represented. So I think this is interesting because it does highlight the fact that not all genes are necessarily the same in terms of their distribution type. What Melindre's gone on to look at then is which genes change from one distribution to another when we look at old versus young, or the treated condition, which is the mice who receive metformin. So what we're trying to do with this framework is to address the next steps of understanding how genes that are changing their distribution, whether they're interesting regulators of aging, whether they reflect greater stability in the older mice, in which case they might be really important for regulating aging, or if they're increased in their variability in the older mice, that might reflect greater degradation of different genes or pathways that are important to healthy aging. And also understanding what is the action of this mysterious drug metformin? How does it bring about this reversal in aging towards a much younger phenotype? And I think for this kind of question, looking at different kinds of distributions allows us that subtlety where we can really pick up on the trends where we don't expect every single cell to be changing, but enough of these cells changing such that that shift in distribution is really meaningful. So I think there's a lot of promise in using this kind of modeling approach. It's certainly a departure, I think, from how people are thinking about this data. So with anything, there's a lot more work that needs to be done, but I think we're finding interesting things along the way. So with that, I think I'm just about to hit an hour. So what I wanted to do is just say thank you to all the people that have really been involved in this work. I ran my group at Einstein, as Miranda mentioned, and was really lucky to have so many talented trainees that contributed some of the work that I've talked about today. They've all gone on to amazing things in their careers, which is really promising, and I think a testament to how dedicated and how talented they are as scientists. I've been super lucky that since being at UQ, I've had so many great people who have wanted to join my group. And in that time, they've done really fantastic work. Melindre and Hui Wen, they are now second-year PhD students. And so the work we're talking about is really only a year, if that, of work that they've done, or a bit more than a year. But nevertheless, they've done really well in the time available. And I'm fortunate for all the support from my postdoc, Dr. Ati Tahirian Fad, and everyone in my group who has really done so well, not just in their own work, but to support each other in doing really great research. I've got some really great collaborators as well who have really supported these different kinds of projects. And as anything, I think we are very interdisciplinary, but we're always seeking new collaborations. And so even on the technical side, I think there's a lot we could be doing that's more rigorous, more sophisticated, more advanced on the statistical side. So if you're interested in talking more, I'd certainly welcome that. That would be really great. We've also been very lucky to get funding from different sources, both in America and in Australia. So we're very grateful for all that that's allowed us to do. So I'm going to end there and open it up for questions if there are any, or general discussion, or whatever you'd like to do at this stage.
Wow.